Today we are going to talk about a very important topic in the field of healthcare and medicine, pharmacovigilance. You might have heard this term before, but what exactly does it mean and why it is so important? Pharmacovigilance, often called drug safety, is the science and set of activities related to the detection, assessment, understanding and prevention of adverse effects or any other problems related to medicine. In simple words, pharmacovigilance makes sure that the medicine we take are not only effective but also safe for everyone. Once the drug is approved, it is used by millions of people worldwide in real life conditions. This is where unaccepted problems may arise. Pharmacovigilance acts as a safety, continuously monitoring medicines to detect this risk and protect patients. Why do we need pharmacovigilance? When new medicine is developed, it goes through years of research and clinical trials. But here's the catch, these trials involve only a limited number of people, usually a few thousands and under controlled conditions. Once the drug is released to the market, it is used by billions of patients of different ages, genetics, lifestyles, and health conditions. This means that some rare, long term, or unexpected side effects may only appear after the medicine is widely used. That's why pharmacovigilance is necessary to monitor drug continuously even after approval and to protect public health. Objectives of pharmacovigilance. The main goal of pharmacovigilance are first to improve patient care and safety by identifying and minimizing drug risk. Second, to ensure public health protection by detecting harmful effects early. Third, to encourage the rational and safe use of medicine. Fourth, to provide accurate and timely information to doctors, pharmacists, and patients. Key activities. So, what actually happens in pharmacovigilance? Collection and reporting of adverse drug reaction from doctors, hospitals, and even patient. Analysis and signal detection, looking for a pattern in the data that suggests a possible risk. Risk management, deciding if warnings, dosage changes, or even withdrawal of a drug are needed. Communication, sharing safety information with the healthcare professional and the public. Pharmacovigilance is a worldwide effort. In World Health Organization runs the International Drug Monitoring Program managed by the Uppsala Monitoring Center in Sweden. In India, we have the Pharmacovigilance Program of India coordinated by the Indian Pharmacopoeia Commission, Ghaziabad. PVPA collects and analyzes adverse drug reaction reports from across the country to ensure medicine safety for Indian patients. Pharmacovigilance is a global effort. The World Health Organization runs the International Drugs Monitoring Program managed by the Uppsala Monitoring Center in Sweden, which collects reports from over 130 countries. In India, we have the Pharmacovigilance Program of India, launched in 2010 and coordinating by the Indian Pharmacopoeia Commission, Ghaziabad. PVPA has more than 250 monitoring centers across the country where doctors and pharmacists submit ADR reports. This makes sure Indian patients benefit from safe medicine. Types of pharmacovigilance. First, passive surveillance means spontaneous reporting, relies on voluntary reports of adverse drug reaction from healthcare professional, patient or manufacturers. No active follow-up is done. Example, area reporting forms in Pharmacovigilance Program of India whose Vigibase global database. Advantages, simple, cost-effective, widely used. Limitations, under-reporting, incomplete data can miss rare or delayed reaction. Second, active surveillance. Here, authorities actively search for ADRs rather than waiting for voluntary reports. Patients are followed up through phone calls, electronic health records, prescription data, or post-marketing studies. Example, cohort event monitoring, post-marketing surveillance studies. Advantages, more complete and reliable data. Limitations, expensive, time-consuming, requires large resources. Third, comparative means analytical studies compares two or more groups to identify association between drugs exposure and adverse effect. Includes Case control studies compare patients with the ADR cases versus those without ADR's controls. Cohort studies follow exposed versus unexposed groups over time to measure risk. Advantages help identify causal relationship and rare ADRs. Limitations requires careful designs may take long time to complete. Thank you.